We're now going to look at nucleic acids. Nucleic acids have three building blocks to them. Phosphoric acid, or you might call it phosphate, a monosaccharide, which in DNA is deoxyribose and in RNA is ribose. And then we have the bases, which are organic amines, and they're going to be ring compounds with nitrogens in them. They're either substituted purines, those are adenine and guanine, or substituted pyrimidines, which are cytosine, thymine, and uracil. So what we're going to do is go through the different building blocks that go into the nucleic acids and what they look like. We have phosphoric acid. It can actually be written as phosphate, and it's considered to be the same thing, or hydrogen phosphate, or even dihydrogen phosphate. They're all derivatives of phosphoric acid, and it's really just basically each of these are phosphoric acid in various stages of deprotonation. We have ribose, which is a 5-membered ring sugar. There are 5 carbons in this ribose. This is actually written as beta ribose. The 2 hydroxy groups are written down, and the methyl hydroxy group is written up. And because this is beta hydroxy, the hydroxy group that is on the far right is also written up. Deoxyribose is the same thing as ribose, but on the number 2 carbon you have the oxygen removed, so it's deox. Now we have the bases. The bases are all aromatic heterocycles that have nitrogen in them, and lots of nitrogen. The purines are adenine and guanine, and they both have this basic structure of a 6-membered ring and a 5-member ring, and they're fused together, and the nitrogens are in their particular places, as you can see. Guanine has a carbonyl in it, and adenine does not. The pyrimidines are thymine, uracil, and cytosine, and they have this 6-membered ring with 2 nitrogens in it. And you can see they all have at least 1 carbonyl in them. Thymine and uracil have 2 carbonyls, and cytosine just has 1 carbonyl. We're going to look at the nucleosides and the nucleotides first. A nucleoside and a nucleotide differ just by a phosphate group. Nucleosides do not have a phosphate group on them. Nucleotides do have phosphate groups on them. So we'll see that they're very similar otherwise. A nucleoside is either ribose or deoxyribose to which a base has been attached. The way it is attached is the hydroxy group that is on the number 1 carbon, the hydroxy group that is beta, is removed and the base attaches instead. If it is a purine base there, then the name of the nucleoside ends in ocene. If it is a pyrimidine base there, then we see that the name of the nucleoside ends in idine. If it's a deoxyribose nucleoside, then you see deoxyribose as the just deoxy in front of the nucleoside name. Here's a adenine nucleoside, and so instead of being adenine, it is adenosine. Now notice, see the number 1 carbon has the adenine attached by one of the nitrogens in the 5-carbon ring being attached to the carbon instead of the beta hydroxy group. If we look at deoxyadenosine, you can just see that the oxygen has been removed. That is the only difference. This is guanosine. Instead of adenine as the base, this has guanidine as the base, and it's again substituted on the number 1 carbon in place of that beta hydroxy group. So guanidine now as a nucleoside is guanosine. This is deoxyguanosine. Notice the oxygen is gone from the number 2 carbon. Cytidine is made of cytosine on ribose here. And you can see again, the nitrogen is attached to the number 1 carbon in the ring. This is deoxycytidine. Uridine is the nucleoside made from ribose and uracil. Now, you don't ever see deoxyuridine because uridine is only found in RNA. It is not found in DNA. This is deoxythymidine, and you see that thymidine is attached to deoxyribose. We only find deoxythymidine in DNA. We do not find thymidine in RNA, and so we don't ever see the thymine attached to a ribose, only to deoxyribose. Phosphoric acid can make esters with alcohols just like carboxylic acids do. And if you see phosphoric acid on the left, and then we see the alcohol there, we can remove water from that to make an ester. And you can see now water's been removed, and so we have a phosphorus oxygen to a carbon chain bond. And this is called a phosphate ester. A nucleotide is just a nucleoside to which a phosphoric acid has been attached as a phosphate ester. So basically, phosphoric acid is attached to the alcohol group on the C5 carbon of ribose or deoxyribose. The C5 carbon is going to be the carbon that is sticking up from the ribose or deoxyribose ring. The way that we name nucleotides is we add a phosphate after the nucleoside name. So this is adenosine phosphate, and you can see that there's the adenine ring attached to the sugar on the number 1 carbon, and then there's a phosphate ester made with the last carbon, the C5 carbon, that is sticking up off the ring. If it is in DNA, then it's going to be deoxyadenine phosphate. If it's on guanosine, then we call it guanosine 5-phosphate. You don't have to worry about the numbers. I'm not going to test you on that. It would just be guanosine phosphate would be the nucleotide. Deoxyguanosine phosphate would be the nucleotide made from guanidine phosphate and deoxyribose. And this is cytidine phosphate and deoxycytidine phosphate. Now, uridine phosphate is only found in RNA, and deoxythymidine phosphate is only found in DNA.